Hello students, today I am going to start the new chapter of class 12th, that is sexual reproduction in flowering plants, which was discovered by Rudolf Jacob Camerarius. Sexual reproduction refers to fusion of male and female gametes forming zygote that develops to form an organism through embryogenesis. In flowering plants, after fertilization, ovary will form fruit and ovule will form seed. Where is the site of sexual reproduction? So, sexual reproduction site is flower. These are the structures of sexual reproduction. During floral initiation, shoot apex get transformed into floral apex. Now we have to understand what is the structure of flower. So what is flower? Flower is the modified shoot modified for reproduction, which considered a compressed shoot in which sepal, petals, stamens and carpels are successive lateral organs which is mean for sexual reproduction. We can understand this with the help of a diagram. So this is the diagram. The floral axis bearing floral organs is receptacle. Four types of organs are present on receptacle in distinct whorl. The outermost whorl is calyx, which consists of sepals, and the inner whorl is corolla, made up of petals. Sepal protects the inner part of flower when in bud. Petals have a great variety of shape, color and arrangement for the attraction of insects. Next to corolla, the inner side is androecium, made up of stamen representing male sex organ. This male sex organ consists of anther and filament. Anther is male organ where pollen grains are formed, and filament is the stalk of male sex organ. This collectively known as stamen. The innermost whorl is of gynoecium, consists of carpel representing female sex organ. It contains stigma, style and ovary. Stigma is the specialized for the reception or the platform for the pollen grains. Style is the supporting tube-like structure supporting stigma, and ovary where ovule develops. Calyx and corolla are known as accessory whorls, whereas androecium and gynoecium are known as essential whorls due to their direct involvement in the sexual reproduction. Now, first of all, we have to study about the structure and development of anther and male gametophyte. So the anther, the stamen consists of two parts, a long and slender stalk called filament. This is the filament. And the terminal bilobed, bi means two, lobe means lobe, bilobed structure called anther. If we cut the TS means transverse section of anther, we will get this type of structure. These are the lobes, and inside these are the pollen chamber where pollen consists. Now, structure of anther. Young anther is four-lobe structure made up of parenchymatous mass of cell bounded by epidermis. So these are the meristematic cells. This is the TS, that is transverse section of young anther. Now it will divide and form archisporal cell and procambial strand. Means this is going in the differentiation, and it will differentiate to form primary parietal cells, primary sporogenous cells inside the layer called epidermis. Now in the mature form it will look like this, and we can understand with the help of this diagram. So the outermost layer is epidermis. Some hypodermal cells inside this, the hypodermal cells functions as a archisporal cell, which are larger, elongated, with dense cytoplasm. This divide to form primary parietal cell and sporogenous cell. Okay, this primary parietal cells to form 2 to 5 concentric layers, and sporogenous primary cell divides to form sporogenous tissue. Now it will form the anther wall. This anther wall will be divided into many layers, in which the first single layer is epidermis, followed by endothecium, and 2 to 3 layers of middle layers, then a single inner layer called tapetum. The endothecium is single layered and its cells are radially elongated with fibrous band of cellulose. These endothelial cells are slightly lignified at maturity. The cells of middle layers are generally short-lived and degenerate at the time of formation of pollen grain. Tapetum is the innermost nourishing layer of anther wall. It is usually single layered and rich in reserve food material. It serves to provide food to the developing sporogenous tissues. They have dense cytoplasm and nuclei. So what are the main functions of tapetum? First is nutritive function. It serves as transport channel through which material reach to the myocytes where the meiosis take place, responsible for producing callase enzyme. For what? For releasing the pollen grain by the dissolution of callose wall. It also helps in thickening of exine and transfer the pollen kit material. Now, formation of pollen grain, or it is also known as microspores. The process of formation of microspore is called microsporogenesis. Micro, you know very well, micro is small, small which is used for pollen grain. Sporo means spore, genesis means formation. Now, in this pollen mother cells, that is PMC, which are diploid cells, that is 2N. It undergoes into the meiosis to form haploid, that is 4 haploid cells, that is N. This is called microspores, which are arranged in the form of tetrad. This division is of two types in angiosperms, that is simultaneous type and successive type. This microspore after division associated with each other for some time, and then this group for some time is called tetra because of presence of four microspores. Now, what happens in simultaneous division? In this, M1 of meiosis is not followed by cytokinesis. Like this, it will not done cytokinesis. See this. There is no cytokinesis, no division of cell. And in successive, cytokinesis occur both in M1 then M2. Okay, so you can understand like this. These are the cells which are dividing according to the different types. This is of successive and this is of simultaneous. Example which is present in successive type are the monocots, and for simultaneous it is present in dicots. This is MMC, means microspore mother cell. Now we have to study about structure of pollen grain or microspore. This is structure of pollen grain. It is unicellular, uninucleate, haploid and spherical. Develops to give rise to male gametophyte. It represents the first cell of male gametophyte, surrounded by two-layered wall. Outer one is exine, which is thick, covered with cuticle, so it is having cuticularized layer. Inner is entine, which is smooth, thin and cellulosic. Exine shows different types of outgrowth containing fatty substance called sporopollenin, which is most resistant organic material. No enzyme can degrade sporopollenin and can withstand strong acids and alkalis. Germ pores are the mark of origin of pollen tube, which are present in number 3 number in dicots and in single in monocots. Some allergies are there with the help of pollen. So in some people the exposure means expression of pollen grains can cause severe allergy or bronchial afflictions disturbance, which leads to chronic respiratory disorders like asthma and all. It can be due to the pollen which fly with wind current. Example is Parthenium. It is also known as carrot grass. Now, some useful pollen products are also there, which are rich in nutrients, specially collected by bees for those honey production. Okay. Nowadays, pollen tablets are also available as a food supplement. It is used mainly by athletes. Pollen viability. So, what is the pollen viability? It is a period during which a pollen retains its ability to germinate. It can be 30 minutes to years, many years. It must be landed over compatible stigma before they lose its viability. Now, the last topic is development of gametophyte before pollination. It occurs in anther, that is microsporangia. In this, during germination, the nucleus of microspore displaced from center. Displaced from center to one side, always to the particular side where the mark of position of germination cell can be done. The cytoplasm become highly vacuolated. Then microspore undergoes many mitotic division and form two unequal cells. The larger part is called vegetative cell and the smaller part is called germinative cell. This vegetative cell has large amount of food reserve and germination give rise to the pollen tube after pollination. And the germinative cell floats in the cytoplasm which further form two male gametes. This generative cell floats in this cytoplasm. Now when the pollen gets matured, they exert some pressure on anther wall and burst and sets free the germinating pollen grains. This uh, process of become freely pollen grains is called dehiscence of anther. That's all for today. In the next video, we will start about megaspore. Thank you.